Morning or good afternoon. Thank you for coming to this mini course workshop. So Bruce Miller, I know him from 2014 when I started working with my own data back in Peru, and he kindly came to provide us this short mini course. He will start explaining us a little bit of how the structure of the course will be. And if you have any questions, we're gonna have a break at around 3 o'clock. Yeah, thank you so much, Bruce, for coming.
Okay. Yeah, I guess in this new age of technology, if you have questions, you're supposed to ask Alexa or Siri, and hopefully you'll get an answer. I have spent, I'm back in the US now since 2012, but my wife and I went to Belize in the late 80s for a one-year project to work on migratory birds. Turns out Belize was a very dangerous place to visit because we got stuck there and it took 25 and a half years before we were able to leave. And the whole time we were doing conservation ecology, initially with small fellowships through Wildlife Conservation Society, and after a few years wound up on staff and running the Belize terrestrial conservation program. I wound up working after we switched from birds, no offense to Betty, to real animals, doing bat work, pioneering acoustic things when bat detectors first started coming on the market for the consumer grade things. And before that, you needed to have really ponderous big equipment and oscilloscopes and all that sort of stuff, which we didn't have funding for, nor any way to operate out in the jungle or the tropical forest. My wife worked on big cats for many, many years, doing camera trapping and working on trying to detect whether or not you could identify individual animals by their tracks. Turns out not, after several years of working, comparing jaguar tracks from Bolivia to ones she knew extremely well and had been following for 3 or 4 years, 5 years where we lived in Belize. The algorithms and discriminant function analysis would just lump them all together and would force an unknown animal, i.e. something from Bolivia, which is a different subspecies, almost twice the size as Central American jaguars, into something that it really wasn't. So we've had a lot of different things that we did there. I wound up really morphing into full-time bat work with the acoustic stuff. And I will go through today. This is not the standard workshop course to teach people how to identify bats per se. Usually those workshops are anywhere from 4 to 6 days, including virtually every night, participants going out and doing their own recording, learning how to actually look at your data and do that. But what has come to light over the decades I started doing this is anytime you're dealing with bats, you're dealing with big data. So I will proceed, and like NPR, this program is brought to you by the Biodiversity Institute, Center for Latin America Studies, Tropical Conservation and Development Program, and a curmudgeon like me. With special thanks to Farah, who, I don't want to say twisted my arm, but who arranged everything and set all this up and is an excellent resource. We're collaborating on bat acoustics now for Peru, and we'll be co-authoring the new fact sheets and interactive identification keys for the vocal signature of bats in Peru sometime in the near future. Why big data? Well, big data relates to data sets that are so voluminous and complex that traditional processing software doesn't really deal with them very well. Big data challenges include capturing the data, data storage, and analysis. And more frequently, paralysis. I've found over and over and over as I've taught acoustic workshops, including since we've been back in the US in 2012, many of them here in the States, that the consultants and state and federal agency people, they put out the monitoring equipment, but then they don't know what to do with it. So much data, what do I do now? Well, hopefully at the end of the day, you'll have a vague idea of at least how I go about dealing with it. So, what's the big deal about echolocation in acoustics? Like all science that builds on the past, a bit of history is called for. When the bat detectors first arrived on the consumer market, a lot of state and federal agencies rejoiced with their monitoring programs. The assumption was by simply hiring part-time temporary students to put on detectors. I have no idea what that was. That they no longer needed to actually hire trained bat biologists to analyze the data or collect it. So there are a lot of agencies throwing detectors out all over various habitats, and then they quickly learned the black boxes were fine, but nobody knew how to deal with what came out of them. So they actually eventually did go back to actually providing jobs for grad students and experienced biologists. This is similar to suggesting in a basic drawing course, that you could master how to draw with little or no experience. The simple two-step process, for an example, how to draw an owl is really good. You start off by drawing some circles, and then it's that simple. So that's kind of like what it is with bat acoustics. It looks simple, but it may be a little more complex than you think. So how does one sample habitats for bats? Traditionally, mist nets, just like birds, have been used for a long time, and there's been an enormous amount of data gathered with mist netting bats in the New World tropics, where I've basically worked.
I'm just going to hide your guest.
Oh, I wasn't sure who was up there. Go away.
Go away. I can't. No, I don't. He looks like I can't hide him.
I will ask him. I'm sorry.
Ask him to turn off his video. Stop video. There we go.
Oh, okay. Well, I'm still standing at the mist net. It takes a long time to get them out. Most of the insectivores aren't really bad. The big frugivores will pull your thumb off if you're not careful. In the New World tropics, there are at least half, if not more, of the species complex that will avoid mist nets. These are all the aerial insectivores, gleaners, what have you, that really have real excellent echolocation abilities. And they, for the most part, avoid mist nets. So harp traps are a whole lot more effective and pick up the whole suite of species that you wouldn't be picking up with mist nets. Acoustic detection provides another and non-invasive method, and it's also very cost-effective. I'll ignore that. Bat detectors, the little boxes that record and detect bats, have been around for a while. There's two main types. There's the heterodyne, which are used mainly in Europe, and that's like your car radio or a portable radio. They're narrow band. They're tuned to one frequency and one frequency only, like your favorite rock station, NPR station, or whatever you have in your car. You're not hearing any other stations that are broadcasting out there, although the airwaves are filled with all the radio signals. So if you're doing bat detection with a heterodyne, you're only going to pick up the 1 or 2 species that are within the frequency range you've got it tuned to. The second one are the broadband detectors that simultaneously will record all frequencies of sound at the same time. And that's what we're going to be talking about. The broadband detectors are further broken into 2 separate categories. There's the full spectrum or time expansion recorders and zero crossing. Many biologists have been using the full spectrum recording, and this is what one of the sonograms looks like. Not quite like an ink blot, but almost. And for full spectrum, as it's generally used, the call pulses, the audio pulses, are sampled at a really high rate using fast Fourier transformation. It actually separates out the various frequency components of the bat call. The output can take several forms. The most common is a sonogram, which we'll show again in a moment. And in the early days, people were so enamored with the technology and the software that they were trying to spit out everything that it could do and present it to their major professor or journal editors for papers and whatever. And these were, the power spectrum was presented as a summation of the various components of the whole bat call. The x-axis is the frequency and the y is the intensity of sound. Well, that's pretty hard to make much out of. And as I'll repeat several times here, people, humans, our brains are really good at pattern matching for visually looking at things. Looking at that, you'd be hard-pressed to match it to other bat calls that you might look at. The sonogram is much better for looking at call shapes and what they actually look like. And this shows a regular sonogram of a full spectrum. Oh, we've got a new guy popped up. In any case, this is a little better to visualize what the calls look like. This actually is the ubiquitous naked-back bat found all over Mesoamerica and northern South America. With full spectrum, you can see multiple harmonics of the signal at the same time. And we'll discuss harmonics and what that means for bat calls a little bit further on. But there's the H1, the fundamental frequency, and H2, which in this case is our dominant harmonic and most regularly seen. In zero crossing, you also will see multiple harmonics displayed, but not exactly at the same time. If you're actually looking, you'll notice that these pulses actually overlap. There's a gap there, but here's the pulse there. That's because this was actually the loudest pulse at the moment, not up here. And then here's the fundamental frequency where there's a few of them were a little louder. But you do see all the harmonics in the zero crossing as well. How zero crossing works? Well, the time for each cycle per second is exactly what the frequency is. That's the definition of a frequency. It's cycles per second, and this is measured in kilohertz. So if the cycle starts here at the 0 line, and then the cycle crosses the 0, going up and down for one complete cycle, you measure that, that gives you, it translates immediately into the frequency of the sound in kilohertz. So it translates very readily and makes a real good, quick and easy means of mapping the bats. This is where early users of the various full spectrum systems were confused in the relation to zero crossing analysis. A lot of them were maintaining, oh, unless we can see all the harmonics, it's not going to be any good. The harmonics are really important. Well, it turns out not so much. In zero crossing, the dots are drawn on the display. This display is the average frequency of the loudest part of the signal. Each dot is measured, and as it crosses the line in the frequency, to actually plot the whole bat signal. And the loudest part of the dot, in full spectrum speak, is this area with the maximum energy. Rather than say loudest, they were saying maximum energy, but I guess, it means the same thing. And again, the cycles, time between the cycles equals the frequency being measured. Here's an example, real quickly, of the dots showing downward curves of a typical Vespertiliana. And as each one of these dots are drawn, it goes through the zero cycle, and it shows the frequency. Along the bottom would be your actual time, and then the vertical y-axis is your frequency. See, these calls, the actual calls, are decreasing in frequency as the dots are being drawn. Again, at any single point in time with zero crossing, only the loudest call part is what's drawn on the screen. If one harmonic is present in the original signal, only one of these can be displayed at one time. You'll notice if you look up above where the red arrow is, there's a gap in the call up above. So the fundamental frequency was just a little bit louder for whatever reason for that one pulse. Very diagrammatic of time versus frequency of an echolocation pulse. We can measure these from the maximum frequency, minimum frequency, duration of each pulse. There's the constant frequency, or CF, part of the call, frequency modulated part of the call where the call is sloping, and then FC is the characteristic frequency, which we'll mention again later. As many adventures in science and technology, with the introduction of new and different types of equipment, there were people who were staunch, almost rabid proponents of their favorite method. Historically, there was beta versus VHS, now DVDs for movies. Music, cassette tapes, Sony Walkman, CDs, DVDs, now iPods, smartphones, and streaming. Technology evolves, and whatever works, works. Well, it may be human nature and not that hard to understand. The mine is bigger or better than yours scenario. I have a feeling if I were to advocate for the Crimson Tide in this crowd, that wouldn't go over very well. Well, I wanted to comment on the great bat detector wars. The literature for a while in the late 90s, early 2000s was replete with about a dozen papers going, literally, a bunch of guys standing around, mine's bigger than yours, or better than yours sort of thing. And there was a lot of papers going back and forth, and I'll comment on one of them in a moment. And mainly this is just to show that if you're going through some of the more, I say, recent literature, I guess for a lot of the younger students, this is old literature now, I guess. But choosing the right bat detector. Well, if it's not full spectrum, it's no good. Oh, well, if it's zero crossing, it does work. On and on and on. The paper on the bottom, the infamous Brock Fenton and a whole host of people published some things which he never retracted, and then kind of corrected himself at one of the meetings, but it never made print. He was talking about, without full harmonics, you can't effectively identify New World bat species. And as I was pioneering everything and testing everything, and I had full spectrum recorders as well. Fortunately, with WCS, we had a reasonable project budget. So dropping $4,500 for a new piece of recording equipment of one of the Swedish Pedersen full spectrum recorders wasn't that much of a stretch. Today I probably couldn't do that. But so I was doing all the recording, both in full spectrum and zero crossing, and asked Brock, I said, well, what's this problem? You were talking about in Costa Rica, you were recording Rhynchonictris, which is a little emballoneurid that flies along and lives along river banks and streams. And you said with the Anabat, the zero crossing, you couldn't identify it because it didn't have all harmonics. I'm really curious if you'd care to share the data. I'll see where I'm missing the boat here because I'm not sure I understand what the problem is. Well, he sent me the data files and I looked at it, and instantly I saw why he was confused. He didn't have Rhynchonictris. He had an entirely different species and a different genus he was recording. He never verified anything. He just set the equipment up in the dark, never watched the bats in real time. So he was recording an entirely different species, a Psychopteryx, which actually has a paired call pulse, which are two pulses following each other that aren't harmonics. It's just part of what the bat does. And he was assuming that that was the Rhynchonictris and it was some magic harmonic that he could see with his equipment that nobody else could. But he never retracted his oops on that, but it still stands in the paper. Anyway, nowadays the bat detector wars are over. Most companies that manufacture equipment offer both because it's clear both are not only accepted as useful, they both have their strengths and weaknesses, and we go on from it. So it's no longer an old argument. It was just during the late 90s, early 2000s, there was a lot of controversy. All by the wayside now. In 2002, Bat Conservation International hosted a week-long meeting that was very interesting, where all of the people doing echolocation research were presenting papers. And this is where Brock kind of said he made a mistake, but it didn't actually get in the proceedings. But there was a lot of discussion, again, about harmonic identification in detail. And aside from all the European participants who were there, who were discussing heterodyne units, and there, they don't have a huge suite of species in most places, in Britain, Germany, Holland, or whatever. And if you only have 4 or 5 species, and their vocal signatures are separated by a reasonable amount of frequency distribution, you dial your thing into your one frequency, as I said, like your car radio, and if you hear it, you know what it is. And the heterodyne detectors actually played the sound back over in human audible terms. So you'd hear kind of tinny sounding, but little smacks and kicks, and you could actually hear calls. And a number of the European bat workers were extremely good at audibly picking these things up using heterodyne detectors. But at the workshop, there a lot of discussion about how much harmonic detail was necessary to identify the bat. Now this is talking about identifying species for ecological and conservation use, not like Elizabeth Calco, who was recording with special equipment that she had from Germany and then videoing in ultra high speed what the bats were doing. And she was experimenting and researching physiology, behavior, and everything else with how harmonics and calls were varying. So that's an entirely different issue, and harmonic detail there was important for those sort of studies. But for ecology and identification of critters, it turns out it's really not that important. A lot of the papers that were presented at the meeting, a lot of the Europeans and the British researchers were using early neural networks to try and identify the bats, early attempts to actually go through and do auto classification. And it was very interesting because they had some really good papers. In this black box neural network, they plug all the acoustic data in and it would sort everything and give some sort of graphic output of what the species might have been. And it looked pretty interesting, but they couldn't ever define what criteria were actually being used by the neural network black boxes, because they have this pruning and all this other terminology where different parts of things drop out and it analyzes different sections. But in the end, at the end of the day, when we sat down over beer after the different workshops, well, what parameters are you really using? I would like to see, so, you know, as we start enhancing our acoustic IDs, we might be able to do it. Well, turns out they're not using any harmonics, no amplitude or anything else. They were using exactly the same parameters that are extracted from the zero crossing thing. So they were spending 3 times as much for their recording equipment than we were and not getting any more data. Anyway, for identification purposes, full harmonic detail isn't necessary. ZC, zero crossing, it's optimized for real-time display of the frequency time characteristics of bat calls, which are what you're going to use to identify your critters. Also, they have very small file sizes. This is management issues are applicable for big data. It doesn't provide the harmonic detail possible with full spectrum. As we've already seen, you can see harmonic detail depending on which pulses are the loudest, but it really provides all the details you need for ID. Back in the early days, we had no choice but to record in real time. You'd sit out in the field with a laptop, and if you were lucky, you got 2 hours, maybe 2.5 hours of laptop battery life. You'd go out into the jungle or some remote area. It took you, a day or so to get out there. You're not going to ever return. You're sitting out there for 4 or 5 nights. So what you do is be real parsimonious with your laptop, and you'd record like mad for 2.5 hours, and then the screen would wink out. You go, okay, do I get excited and put in a new battery? Or, well, we still have 5 more nights to record, so you were done. We kind of lamented that we had to sit and record in real time, feed the mosquitoes, recycle our nutrients, and all of those wonderful things. But the few of us that were doing the early pioneering stuff, recording in real time, opened up everything we needed to learn about what the bats were doing. You're sitting there in real time with spotlights, you're watching the bats, you're seeing multiple bats come through, you're learning about the ecology of the bats. We weren't setting a black box into the field, going back to have rum and cokes back at the camp, and then go back to pick it up 3 days later. We're actually out there, which is what field biologists do. I mean, a little rum is probably not bad to prevent malaria, or actually you need quinine water and gin and tonic. But in any case, the real-time recording was a real benefit because we learned an enormous amount for the first 8 or 9 years, even when we could record passively, remotely, we continued to record in real time. When I began the first field evaluation in the New World tropics, every recording I had resulted in an unknown species. We had no idea what any of them were. Nobody had recorded them. It was clear that every call sequence that popped up on our screen in real time, we had all different shapes of calls, call frequencies. We had initially, well, was this one guy, several guys, different families, different species, different genera? Took a while to figure out. But the moment you turn on the equipment in the early days, it's like, wow, pretty neat looking stuff. Let's see if we can figure out what it is. Well, the first task was to match faces with voices. That meant we had to capture bats. And again, mostly harp traps because most of the echolocating guys avoid mist nets, and there wasn't any way to match faces to voices with mist nets. We needed to guarantee that no other species were flying around in the dark to inadvertently be recorded. I mean, we did subsequently have bats that we hand released. You turn them loose in the dark, and record them. And again, how do you know what you're recording is the guy you released versus the 3 guys that have been flying around here for 20 minutes that might be completely different? Well, we had to release them sometimes under very highly controlled conditions in our acoustic laboratory. And you can imagine that in Belize. Being conservation biologists, it doubled as our kitchen. In the field, we use light tags, and those work pretty well. A lot of times you'd light tag a bat, you'd follow the little light going through the dark, it would land in a tree, and all of a sudden you'd see the little light tag just drop to the forest floor, then you'd hear the bat fly off, and you go, okay, where did he go? Where did he go? So that worked mostly, but not always. Other methods were tried, zip lines, where you had a real heavy piece of monofilament fishing line struck up maybe 100 meters, 50 meters, and then you put a little lasso or whatever around the bat's foot, that you could release. Somebody would go, okay, release, and they'd let it go, and so the bat wouldn't fly off. And in theory, he'd run up and down. And then also Jose Ochoa, one of my colleagues in Venezuela, started using the comet method, where he just put the little lasso on the bat's foot with a long line and let it circle around, and also extensive roost recordings. Well, we found out that most of these calls were not useful for identification. They all emit generic clutter calls, which we'll talk about a little bit later. And I guess if you were at a karaoke bar and somebody tied a rope around your foot and started swinging it around the room, you probably wouldn't sing very well either. So, it was highly disruptive to the bat. And, no bats were hurt in these trials. They were all released, and they were just kind of, okay, let me go. But you don't always get really good calls that way. Roost recording, you can get calls if you understand physics of sound and what the bats are going to do. And if that open doorway were a roost space, if I went over towards this back table and tried recording, I would get generic leaving roost similar to clutter calls, which we'll show examples of, and you're not going to get anything. If I went out into the corridor or the place outside, it was 50, 60 meters away. As the bats came out, they'll actually shift from a clutter call, navigation call, to a search phase call, which is diagnostic. And you can frequently get calls from roosts, but you have to be real careful about that. With experience, it also became really clear that the vocal signatures, as we started calling them, which are the search phase calls, have both family and genus-specific traits. We tested this, Mike O'Farrell, Jose Ochoa in Venezuela. We went down to some of the conservation protected areas in Venezuela, where Jose was with Wildlife Conservation Society as well. And we recorded all over, 10, 12 protected areas, and also continued to capture bats with harp traps. But we started seeing call signals that were very similar to things we knew, but they didn't match any of the frequencies that we had seen. One of them is we're sitting at one of the first courses we did down there. The acoustic workshop was at a university, and the students had little or no funding, which surprised students. Always have lots of funding, right? So we couldn't travel a long distance from the workshop to do recording, so we went out to the botanical garden, literally outside the lecture hall that night, to set up harp traps and try and do some recording. And we put up one of the harp traps between a couple of trees, so there was a little bit of a corridor effect to catch something. And I was sitting about 100 meters away with the laptop. Again, we're recording in real time, and the calls start scrolling across the screen, and I'm going, that looks like the bulldog bat, the big noctillionid leperinus, the fishing bat, but the frequency is way, way higher. That can't be. I wonder if it's the smaller species, because the larger the bat is, generally the lower the frequency. The smaller it is, the higher the frequency. It's a size ratio. And we were saying, well, puzzling. That could be the other species of Noctilio. There's only 2 species in that family. And almost instantly, the calls went off the screen, no longer had any signals. I went, well, there was our chance. And about that time, one of the Venezuelan or Colombian students, I don't remember which, started screaming, we got a bat, we got a bat. So we trundled down to the harp trap, and there it is, Noctilio albaventris, the smaller guy. So we verified the other species. And as we continued our field work with a host of other species and other families and genera, we would hypothesize, we knew what this big bat was, it had a big forearm, and we knew what the little guy was, but reportedly in the area were a couple of intermediate sized species in the same genus. So we thought, well, if this is holding up, then the calls of these other guys that we don't know yet should be intermediate. And sure enough, as we were recording, we started recording these other species that we hadn't had verified before. And that's important because I'll talk about sono species that we can't identify yet. Anyway, acoustic techniques also provide insights on how habitat is being used for ecology. When you have the feeding buzz, which is this stuff here, this is the sac-wing bat that I was talking about with the typical paired pulses that Brock thought was harmonics of Rhynchonictris. But as it shifts to a feeding buzz, it's quite distinctive. Here's another moacid feeding buzz. So with acoustics, you can actually tabulate how the bats are using the habitat. If they're just going from point A to point B, or if they're actually making their living there and foraging. And bats have bimodal ecology. They have a roosting ecology and a foraging feeding ecology. A lot of times they're roost specific and they have to live in one area if that's the only place caves are, but there may not, or hollow trees or wherever they might be, but they forage somewhere else, maybe 5-10 kilometers away. So this gives you an idea of tabulating how the bats are actually using the habitat. If you're developing a protected area, do you need to only protect the roost area? No. If they make their living and get their food 15 kilometers away, it's kind of like some of the early Quetzal reserves for birds in Central America. They reserved all these high elevation areas because that's where they saw the Quetzals. Well, that was only seasonally. They'd migrate down the mountainside to areas that were getting ravaged by forest loss. So you have to be real careful when you're looking at where critters are for conservation. But feeding buzzes and acoustics can help you look at some of that. So acoustic methods are effective for assessing and reassessing rarity. Everywhere I've gone teaching workshops in the New World tropics, and we've turned on the equipment, we've picked up probably anywhere from 3 to 10 new distribution records, because these are species that avoid mist nets. They're not in the museum collections, and almost none of these areas that we worked had ever been harp trapped. So it was all new distribution records for that. Great for surveying protected areas, species habitat use. You can use the data to determine habitat preference. Fixed site monitoring, active, real-time monitoring. Passive, set the equipment out, go back in 10 to 14 days, pick it up, and you've got big data. Transects, you can do recording driving, walking, in a boat. Real super way to do river transects. You just start at one end and you at a certain fixed speed and record everything going along. It's really, really powerful. You can't miss net or harp trap moving down the river, but you sure can do the acoustics. So bat conservation essentially needs to be considered in the context of landscape ecology. Without adequate distribution data, habitat usage, we can't even begin to manage protected areas. So acoustic methods have provided new insights that weren't previously possible. How and where you record will influence your results. If you're in an open area like A, where you can pick up some understory bats, but it's also open canopy, you'll pick up the low or high-flying Diclodurus, the big sackwing bats, and the high-flying mollusids, as well as understory species. If you're just in the forest with closed canopy, the only thing you're going to record are the understory species. That doesn't mean that the mollusids and the Paroptrix, the little sackwing bats, and the other high-flying bats aren't there. It's just the sound isn't going to penetrate the canopy. So you need to be aware of where you're recording and what your results are telling you. If you're in an area like C, with what we say broken canopy, like palm trees, of course, all of you have seen palms, they're open, so the sound can actually penetrate through the leaves partially. You'll get broken sound through there, so you will pick up some of the high-flying species as well as species that are out over the open. And in C, that's over an open waterway, and you'll pick up actually echoes of the calls over the water that aren't actually part of the regular bat call that might confound your identification. How long you survey and when will influence your results. To characterize common species within a community, a few hours at only one location, not going to cut it. Minimum three nights, dusk to dawn. Better, 5 or more nights, dusk to dawn. To detect really rare species where there's only a couple of individuals, you may need 7 to 10 nights. You need to be aware of lunar cycle bias. Bats do avoid flying in the open during full moon. They're still flying because they've got to eat and drink, but they stay more hidden, of trying to avoid owl predators and other things. So be aware of lunar cycle bias. Weather bias, we've had some of the best bat recording and capturing we've ever had in areas in the tropics where you have a late afternoon rain and then it clears up and you have a flush of insects that are just everywhere. And same thing with watching swallows or other birds foraging just at dusk over grassy meadows and everything to pick up the insects. The bats exploit that as well. A couple of papers I'll review. Moreno and Hafner in 2000, they actually looked at completeness of biodiversity inventories, and they were showing that about 14, 15 nights before your species accumulation curve starts flattening out with asymptote. So you will get a real steep accumulation curve at the beginning, and then it starts flattening out, but you may still be picking up species further out, a few more nights out with more rare species. Goranson and et al. In an older paper were talking about detection capabilities of bats. They're saying acceptable accuracy wasn't attained for at least 15 sites or 15 visits to detect probability of occupancy. It says it's not even certain that species, even if they're present, that you're always going to detect them. A lot of this has to do with physics of sound, how close the bats are to the detectors. The detecting ability diminishes with distance. Also variables like I showed in the little graphic, vegetation, clutter, canopy closure. Just be aware that there is some bias in your acoustic data as well. You're never going to get everything. At the end of the whole presentation, what I can do so people don't have to scribble down the references, I can convert all of these PowerPoints to PDFs and give them to Farah so you can actually have copies of this stuff if you wanted it. Additional points, Waller and Baldwin, and this was relevant for Florida, for the Florida bonneted bat. and I started working on Florida bonneted bats and working with acoustics in 2014 and 2016 down in the Seminole tribe area, way in the southern part of the country or state. But detectors at high elevation were much better detection probabilities for low frequency bats like the UMOPs, the Florida bonneted bat. So with that in mind, we'll go on to the fun and games. So bat party games for pin the tail in the donkey, plug their ears. The sheer volume of acoustic data can be daunting and nearly paralyzing if not well managed from the beginning. A lot of the early wind turbine acoustic work kind of boggled my mind. It's not unique to WCS where I was, but with the likes of George Schaller and others mentoring and dictating and driving the WCS field research programs for conservation people. If you weren't doing good science, you weren't part of the team, and there wasn't much value in you even being in the field. So I was shocked to read a lot of these early reports about wind turbines. These people were getting inundated with full spectrum acoustic data, and in the reports they chose to only analyze subsets of the data, 15 minutes out of each hour. Chances are they're going to miss a whole lot. I maintain don't throw out 3/4 of your data. You need to look at everything. You never know. The single file you skip over may be a new record for your distribution area. So the goal of this mini course is to discuss ways to work smarter, manage and analyze acoustic data more efficiently. And with that, I will break for this one, take a drink of water, and if anybody needs to stretch for a moment, I guess that's possible. I'll get the next one queued up. And there will be plenty of time for questions. Come on. My little laptop is more of a notebook tablet than it is a laptop. Okay.
Can I ask a dumb question? Somebody that doesn't
No dumb questions. Go ahead and ask.
So when you're talking about identification, you're only talking about species identification, not individuals, not of individuals, like individual bats.
Yeah, you can't identify individual. You can identify species. There's no difference in the calls within actually the species themselves. There we go. Took a long time to come up, but yeah, no individuals, just your species, genera, families.
Because if you could do the individuals, then you could do some kind of like catch and release.
Right.
Studies.
No. Yeah, I mean we've done huge numbers of bats recorded, and you don't really see enough differentiation for that. I'm sure the bats can figure it out, right. But yeah.